before giving the vaccine to a human population, you need to know the efficacy of vaccine. Okay, so you give this vaccine to some candidates. Okay, in those times, now we have ELISA and everything, we have many immunological techniques. In those times, suppose you didn't had any of these immunological assay techniques. So suppose you want to test whether your vaccine is efficacious or not, how will you do that? Suppose I injected you with a polio vaccine, okay? Your body is making antibodies, okay? But her body is not making antibodies. So your body is high in antibodies, your body is low in antibodies, okay? So what I'll do, I'll take a mice, okay? Inject it with virus. The mice will become infected, okay? At the same time, I will inject his sera. His sera should contain antibodies related to polio. Okay, if his vaccination is successful, his sera should contain antibodies or not. Okay, so at the same time, if we give this sera, his sera, your name is Nitikesh. So Nitikesh sera to mice, will it able to neutralize that particular virus or not? Okay, so that mice will be live. So that is called mice serum neutralization test, very easy. But if we give your sera, your name is Smriti sera, and if the mice dies, then we can be sure that her blood doesn't contain antibodies. Okay, that is a very simple test. You know, you don't need this modern infrastructure. You need a good thinking to become a good scientist. Okay, so many discoveries. You know, Edward Jenner, how he discovered this vaccine. Okay, a small observation. A milkmaid, smallpox. Okay, so milk. You know, there is a disease in cow called cowpox. So what he used to observe that the milking maids, okay, who milk the cows and cows who are affected with this cowpox, those milking maids were not affected with smallpox that time. Okay, and entire population smallpox was very much relevant that time. The observation what he made, that yes, there is some other type of, you know, this thing virus which is related to this smallpox. Because of that, these milkmaids are not affected with smallpox. Okay, a small observation. So you need not have a huge setup to become a good scientist. Your observation is very important. Day-to-day observation is very important. Okay. So these are some kind of means. See, this is a very simple example. I wanted to tell you how these preclinical things work. What is the importance? We never know. We make vaccines, okay, but we never know what is the story behind these vaccines. How we do different types of safety tests. How we do different type of efficacy tests. And once this drug is proved, a drug or a vaccine is proved to be efficacious, to be safe and efficacious, then it can be taken to the clinical trial. So this is the whole story of a vaccine, how it goes from a, you know, lab stage to a preclinical stage and then to a clinical stage. See, preclinical testing, again, I told you it seems to be very easy, but it's difficult. It's difficult. 95% of the times we fail, I told you, and the reason for failure is because, you know, in mice or rat in preclinical tests we get very, you know, encouraging results. Okay, scientists sometimes they jump on their table. Okay, we have developed a vaccine or we have developed a drug which totally eliminates this tumor or a cancer from mice, but when it goes to clinical stage they fail. Okay, the reason is we don't choose good preclinical models. We don't do our homework. Okay, that is what I want to emphasize. Preclinical testing, लगता है कि okay, we'll test it in mice, we'll test it in rat, but you know, you need to make some good efforts. You need to identify good models. When you do wound healing, you know, diabetic wound is a big problem. Why diabetic wound is a big problem? Diabetic wound doesn't heal. Okay, so many times due to diabetic wound, you know, foot has to be amputated, hand has to be amputated. Okay, so making a diabetic model in mice and rat, it's very difficult. Very difficult because in mice, the skin heals by contraction, not by epithelialization. जब आप लोगों को चोट लगती है, whenever you are, you get a wound, your wound heals by epithelialization, not by contraction. Okay, epithelialization means new epithelial cells will migrate to the site wherever wound is there, and it will heal up. Then collagen will be laid, then strengthening will be done, and wound will be healed up. In most of the animals with loose skin like mice, rat, and even dogs and cats, in horses also, and even in our neck area. Okay, in our neck area, wound heals by contraction, not by epithelialization. Contraction means you have gone to a doctor, you have seen, when we have a big wound, okay, a big injury, we go to a doctor, doctor sutures the wound. Okay, why does doctor suture the wound? Otherwise, it will heal by secondary intention, it will take a long time. When doctor sutures the wound, it heals quickly, okay, because two ends of the wounds are opposed to each other. Okay, so same thing happens in animals. Whenever in mice or rat, whenever there is a wound, there is a small muscular layer beneath the skin. उसको बोलते हैं paniculus carnosus. There is small muscular layer, paniculus carnosus. So as soon as there is a wound in mice or rat, that muscle contracts, and the opposite end of the wounds, they come to opposite end of the skin, they come to close to each other. Okay? So when they come close to each other, wound heals by contraction. Same thing happens in our neck area also. In neck area, we have a muscle. Whenever there is a wound in our neck area, that muscle contracts. In our neck area, the wound heals by contraction. But in all other parts, wound heals by epithelialization. So when we have to create an animal model for wound healing, mice and rats, they are not a good model. Actually, pig is a very good model. Pig skin morphology is very much similar to that of human beings, but again, doing experiment with pigs due to ethical issues and due to space constraints, it's very difficult. Okay? Maintaining a piggery is very costly. Okay, again, getting ethical permission also becomes costly. But in mice and rats, we can create a model which heals by epithelialization. And how do we do that? We put up a splint. We create a wound in mice, and we put up a splint, a silicone splint on the corners, due to which wound is not able to contract. Okay, so because wound remains in an uncontracted form, again it heals by epithelialization. Okay, so that is how we create different types of models. So that is what I am saying, preclinical testing is not easy. But again, 95% of the times we fail. But still, a very astonishing fact is that most of our biomedical discoveries, they have been possible because of animal testing. Most of our biomedical discoveries, be it cancer, be it diabetes, be it cardiovascular research, most of our biomedical breakthroughs, all those are possible because of this animal research. And you will be astonished to know that 96 Nobel Prizes in physiology and medicine, they have used animals. Only 12 are there which have not used. Even 2023 Nobel Prize for this mRNA vaccine, we'll talk about that. They again used animals. So there are many groups, many scientists, they have been able to give good results, breakthrough discoveries only because of use of these animals in biomedical research. There are few examples where preclinical research and vaccine development has used animal models. You being pharmacist, now in bee pharmacy, sir, their animal experimentation is mandatory or not? Not. Software based. All the experiments are now software based, but in MPharm, research can be. And in MPharm, okay. So being a pharmacist, definitely, it used to be a very important part of course curriculum some 10 years, 15 years ago. Every pharmacy college used to have an animal house facility, and it was mandatory actually. But now, this ethics, they come into play, UGC and Pharmacy Council of India, I think so, they have banned the use of animals for undergraduate courses, but not for MPharm, I suppose. Yeah. So there are few examples where these animals have played a very important role. The one I told you about, this mouse and monkey. So there was a scientist called Armstrong. These mice make, if you grow any virus or if you make a disease model out of mice or rat, life becomes very easy. You know, working with monkeys, sheep, and horses or pigs, it's very difficult. These are large animals, maintaining them, doing research with them. There are many ethical issues. Okay, ethically getting permission to work with large animal, again, it's very difficult, very very difficult. But if you can create a mice model, means you will benefit a lot out of it. But developing a mice model becomes difficult because chimpanzee, monkeys, they are more closer to us. And though our genetic makeup with mice and rat is also very much similar, we share more than 99% of the genes which are similar. But still, there is a difference. There is a difference. They are distant from us. But if you develop a mice model, your life becomes very easy. Very easy because in a small room like this, you can, rear up to something around 20 to 25,000 mice. Generation time is very less, 21 days only. Okay. So, in large animal, this generation time is high. So in mice, only 21 days. You get next generation in 21 days only. So again, developing a mouse model for polio vaccine, it was a challenge which was taken up by a scientist called Charles Armstrong, and he injected this polio virus directly into the brain of mice. And I told you, if you inject a polio virus through oral cavity or through nasal cavity, or even if you directly inject it into the limbs, the mice won't develop polio. But he injected this virus directly into newborn animals, and he was able to develop a very good model for this polio. And again, monkeys were extensively used for culturing this polio vaccine and testing these polio vaccines. Testing and culturing, both monkeys were very much used. So because of this, we were able to develop a mouse and a monkey model. And because of this, millions of children are today able to walk on their feet, because this polio was a dreadful disease long back. So again, the role of animals. Yellow fever, very interesting. Yellow fever was a big killer in those days. In 1940s, yellow fever was a very big killer. It used to wipe out populations. And this Dr. Max Tiller discovered that when virus was passed repeatedly through mice, it weakened. I told you how to make a virus weaker. You put it into a new environment. Okay, so what he did, he put the mice into mice and then multiple times passaged it. Means he injected into the mice, isolated the virus, again injected, again isolated. So that made the virus weaker. And he was able to develop a very famous vaccine called 17D vaccine. And this work earned Thaler the 1951 Nobel Prize for medicine, the only vaccine-related Nobel Prize ever awarded. Only vaccine-related Nobel Prize ever awarded was in 1951 to Thaler. You may argue that in 2023 this mRNA vaccine also got a Nobel Prize, but that was for a technology, mRNA-based vaccine technology. That was not for a COVID-19 vaccine, but for a vaccine technology that led to development of COVID-19 vaccine. This is the only Nobel Prize, yellow fever 17D vaccine, given to Thaler in 1951. Only vaccine-related Nobel Prize ever awarded, and a mice helped to create a vaccine that saved millions of lives. Again, very interesting story. 2023 Nobel Prize. I told you it was given to a technology, not to actually this COVID-19 vaccine, but to mRNA technology which led to development of COVID-19 vaccine. All of you know about mRNA vaccine? Okay, so mRNA vaccine is very interesting, and this is a new concept. Crisco and Westman, they were awarded Nobel Prize for this technology. But viruses, there are two types of viruses, DNA virus and RNA virus. Okay, So if you inject a foreign RNA into a human being or an animal, it may identify it as a virus, okay, and it may elicit a huge immune response. Whenever we get higher viral disease, sometimes our immune system becomes our enemy. Sometimes cytokine response is so high that even it leads to death. Sometimes cytokine response is so high that even it may lead to death of that particular animal or human being. So the challenge with these two scientists was that normal mRNA, though a very good vaccine candidate, okay, mRNA integrates into the body and produces some proteins. Proteins are antigenic in nature, which in turn makes immune response, okay, immunity within our body, antibodies within our body. But again, same time, if a normal mRNA is injected into mice, what they observed, if a normal mRNA is injected into mice, though mRNA was a good vaccine candidate, but when normal mRNA was injected into mice, it used to produce huge inflammation in mice. Huge inflammation that, even it was fatal to the mice. Okay. But then what they did, they modified this mRNA. They discovered that by modifying one base, pseudouridine, okay, so by just modifying one base, this immune system no longer recognizes mRNA as a threat, okay. So when our body is able to identify a foreign antigen or a foreign virus or a foreign bacteria, it elicits immune response. But if our immune system doesn't recognize it, so that was the turning point. They just changed one base and then again tested it into the mice, and again modified mRNA showed no inflammation, and they were able to develop a very safe vaccine which gave us a COVID-19 vaccine, a technology which gave us a COVID-19 vaccine. And now multiple work is going on mRNA vaccine. In few years you will see many different bacterial viral diseases with this mRNA vaccine coming into the markets. In COVID-19, we had to hurry up because that was a pandemic. But usually a vaccine development or a drug development takes something between 10 to 12 years. It takes a very long time. That is the minimum time what I am telling you, 10 to 12 years. It may take even longer. Billions of dollars of investment, but it may take even longer. But then now in future you definitely will see many drug candidates, many vaccine candidates out of mRNA coming into the market. Again, before closing my talk, just I wanted to tell you the story of vaccination. How animals, they play a big role. How you people, after the vaccine is developed, play a big role. Again, I told you, without your contribution, making any eradication program won't be possible. So, going by today's theme, pharmacists as advocates of vaccination, pharmacists help turn scientific ideas into safe vaccines. So many of you will be definitely, I know many, there are people in our institute also who come from a B Pharma background, and they are faculty, they are doing, good work in different areas. I know many people from B Pharma background who are very good scientists in very, even they are very good scientists in basic research institutes. Ours is a basic research institute, okay. So in NISERS, which are primarily basic research institutes, you will see many people from B Pharma background. So pharmacists help turn scientific ideas into safe vaccines, because today's theme is vaccination. So safe ideas into safe vaccines. So pharmacist as a scientist can turn scientific ideas into safe vaccine, again by guiding dosing, formulation and preclinical testing. Many pharmacists, many B Pharma, M Pharma people, they are into this preclinical testing. I know means the industry, many B Pharma, actually, companies, they prefer this B Pharma, M Pharma people with animal testing background, who have good amount of experience in animal testing. They prefer them like anything, okay, because you have a good knowledge about drugs, you have a very good knowledge about regulations related to these drugs, and again if you have a good knowledge about this preclinical testing in animals, that becomes a very good added advantage. It gives you a very good edge in the industry. I know many people, many friends of mine, they are doing very good. They are in top positions. Formulation again, formulation in older times, there was a lot of work done by pharmacists in this formulation and now also. And again, dosing. Okay, so I told you, gave you many examples related to vaccine administration, what challenges you may face, how each and every individual is different. So means if you have been given a task of mass vaccination, again each and every individual is different. Each and every individual may produce different kind of immune response. Okay, so your duty as a pharmacist to ensure that each and every individual is protected from disease whenever you run any eradication program. So onus lies on you, and it is your week. So definitely, I'll give a big credit to all the pharmacists, and I will like you applaud yourself. A big clap for all the pharmacists. I'm happy to take any questions. Not only related to vaccine, or if you have any doubts related to preclinical testing, animal testing, different animal models of disease, and if you have some research idea in your mind, definitely now you can ask me, or even sir has my contact. Definitely after this event also you can contact me. If you have any questions, please.
So, I have a question which is related to vaccines. I initially knew about the only attenuated vaccines, not the cured one. So, I would like to understand more about the cured one. On how is it, like the antigen, is it going to boost the immunity of the body? Yes.
See, very apt question. Attenuated virus are live viruses. Okay, they are able to produce a very robust immune response. So the amount of antibodies what you get means the immune is for long. Means there are certain vaccines which are given once in a year, or there are certain vaccines which are given only once and immunity remains there for lifetime. But there are certain vaccines which you have to take every year or every 5 years or every 10 years. Live attenuated viruses, they give very good robust immune response. The memory is also very good. Killed vaccines, they have an advantage. Killed vaccines means you will kill a vaccine, but still antigens remains there. Okay. So because body is able to identify the antigens, there are different types of antigens. Even if killed vaccine has a good epitope. Epitope means the site where your immune system will come up and identify that particular epitope, that particular region in the antigen, and then give you an immune response. So definitely in killed vaccine also you get a immune response. There are many examples of killed vaccine, and there is a very good advantage. Okay, so suppose in a country which is already free from a particular disease, it is always recommended to use a killed vaccine rather than a live attenuated vaccine. I told you live attenuated vaccine during multiple replications, when you give a live virus to a particular human being or particular animal, during replication it may re-originate, means come back to its original pathogenic state. Okay, it may infect. But if your country is already free from a particular disease, it is always safe to give a killed vaccine. Okay, so killed vaccine also is effective. It depends upon the type of antigen you are using, or means more refined a vaccine is, better immune response, better specific immune response it gives. Definitely killed vaccine, polio, we have both kinds of vaccines. Polio killed vaccine is also there. The first the polio vaccine was made by Sabin and Stark, है ना? Stark and Sabin, है ना? So the first polio vaccine actually made was a killed polio vaccine. Okay, they killed the polio virus using some chemical, I think so, formaldehyde. The second polio vaccine which was made by Sabin, actually it was the live attenuated polio vaccine given through oral route. Okay, so it was found that through oral route, when it gives, it mimics the normal, pathway of this virus, and then able to replicate and elicit immune response. But again, at the same time, that virus is not able to affect the nervous system of the human being. So in polio, you have both kind of vaccines: killed vaccine also and live attenuated vaccine. The difference is, in killed vaccine, you are killing the virus altogether, and in live attenuated vaccine, you make the virus weaker. Okay, so both have some advantages or disadvantages, but primitive. These both vaccine, killed and even live attenuated, both are primitive vaccines used since ages.
Yeah, but
Sir, you told me that in the ancient time, scientists talked and developed the kid vaccine for polio. Sir, my question is, how can they be so sure that the virus is killed? It maybe the virus is getting weaker. So if I take the virus and inject it into the another mice brain, then no paralysis occurs, or mice behavior pattern does not get changed. So how can they be so sure that the virus is killed, not gotten weaker?
See, actually, virus is very fragile outside the body, very fragile. Okay, so it's very easy to kill it. Yes, there had been accidents in this polio vaccine also. There had been accident even with killed vaccine also. If you go into the history, there had been accidents. There was a manufacturing plant, means where from somehow they were using, I think so, formaldehyde also only for inactivity. But there were two batches of vaccine where somehow this virus was not killed, and it accidentally got injected into the human population. But again, then you have to come up with better stringent quality assurance and quality control measures. Okay, so better, more stringent quality control measures. You know about thalidomide tragedy. Thalidomide tragedy. Okay, so after that, the drug testing rules regulation altogether changed. So thalidomide was a drug which was given to treat morning sickness, and especially in pregnant women, pregnant ladies. This drug was prescribed. It was approved in US, but not approved by FDA in US. Okay, but then this drug was rampantly prescribed for treating this morning sickness in pregnant ladies. But then after when these children were born out of those pregnant ladies, they had some limb defects, congenital defects. Okay, so when these everything was traced, it was traced that this drug is responsible for those birth defects. But then this drug was found to be safe. Even in preclinical testing in different animal models, it was found to be safe. But again, when it went to human population, so better stringent, more stringent quality control measure, that is the only way we can ensure the safety of the drug. There is no other measure.
receptors including
Anyone else?
receptors and factors are for
Okay, fine. Okay.
Good afternoon, sir. It was indeed a wonderful session. I have one question. In which condition we use mRNA vaccine rather than the traditional vaccine which are available in the market?
Actually, I am not the right guy to answer this question, but I'll try to answer your question. More purified the vaccine is, if you give in killed vaccine, okay, you give whole virus. Each and every part of virus, or in killed vaccine you give whole bacteria. Each and every part of bacteria or virus is not a antigen which is able to elect the immune response. The more purified proteins are good antigens. Okay, carbohydrates are poor antigens. Okay, a bacteria contains some carbohydrate entity, some protein entity. If you give a particular vaccine in a more purified form, if you identify a protein which is actually electing, which is giving an immune response in that particular vaccine, if you inject that purified protein, you will get a very specified immune response. The immunity will be there for a longer period of time, and the chances of side effects, okay, the hypersensitivity, all those will be very less. Protection and efficacy will be better. Okay, so more purified a vaccine is, more specific a protein is, definitely you will get a better immune response. In case of mRNA vaccine also, the protein which is produced, actually it is very specific, very specific. Okay, so again, mRNA vaccine, you can say why it is better as compared to these traditional vaccines, but it may depend upon the candidate to candidate different. But why it may be better? Because it may give rise to immune response which is very specific. Okay, it leads to production of some antigens which are very specific, and we know the coding for that. Okay, if you know the coding for that, it will produce that protein which is very specific. Again, your immune response will be very specific. Okay, immunity will be better and the memory will be better. So definitely, they have advantages. But again, I told you this in mice previously. RNA, RNA virus. There are some RNA virus, some DNA viruses. So even injecting RNA or DNA may lead to some type of hypersensitivity, overreactivity. But again, they changed one of the base, actually, after which this technology came into existence. So definitely now work is going on. It will be more specific, better.
Okay. So I heard about healing of the wound, like healing of which is related to diabetes. Yeah. Like, how is it linked diabetes to the healing of the wound, like healing pathway? How is this linked?
Very good question. Diabetes is a disease which leads to a silent death. If you ask any clinician, whenever heart attack is there, a person gets some symptoms of heart attack. Okay, he gets some symptoms. What are those symptoms? Nervousness, high heart rate, sweating, and then pain in the chest. But in diabetic neuropathy, what happens? Diabetic is a very, very disturbing disease. It kills you in a way you won't even come to know. Okay, diabetes leads to diabetic neuropathy. First thing. Okay, so your neurons becomes insensitive. Okay, they won't communicate the message to your brain. Each and every cells require glucose energy. Okay, so when your cells, they become insensitive to insulin. Okay, so uptake of glucose becomes poor. So each and every cell of your body is affected. It can be your immune cells, it can be your neurons, it can be your normal epithelial cells. Each and every cells of your body is affected. Diabetes puts a lot of pressure on your kidneys. It puts a lot of pressure on your neurons. So definitely, first thing is diabetic neuropathy. Whenever you get a wound, suppose in your feet, okay, first thing is you won't be able to come to know that you have a wound. It may become worse, that wound will be increasing, but because you have a neuropathy means pathological condition of neurons. So your brain won't sense you have a disease or a wound in your foot. So that, because pain sensation is less, so wound will keep on increasing without your body knowing. In heart attack, what happens? A diabetic person, when he gets a heart attack, there is no pain, there is no classical symptoms of heart attack, and a person is sleeping and next morning he dies due to heart attack. No one comes to know. A normal patient will be, when he gets a heart attack, you see all the symptoms, all the classical symptoms of heart attack. Okay, same thing happens in diabetic neuropathy. You won't come to know that there is a wound. Your brain won't be able to sense due to neuropathy, first thing. Okay, so your wound will keep on increasing. Secondly, again I told you each and every cell of our body, it requires what it requires. It requires energy. Your brain cells require a lot of energy. Your immune cells require a lot of energy. Your epithelial cells, each and every cells. Okay, and because due to insulin insensitivity, the glucose metabolism is affected. Each and every cells to divide to replicate, they require glucose. They require energy. Okay. So because each and every cell is affected, definitely they are not healthy. Your epithelial cells are not healthy. Your immune cells are not healthy. So it takes a lot of time for a wound to heal. It heals, but it depends upon your condition, your blood glucose level. If you are able to maintain, yes, it heals. But sometimes it progresses to a point that you have to amputate the foot. There is no other option. So it is multifactorial. Okay, but one of the reasons is this diabetic neuropathy. Due to diabetes, neuropathy is there. Your neurons will become neuropathy means pathological condition of neurons. Okay, so neuropathy and then your immune cells and your normal epithelial cells, they don't heal. They require glucose. That is the reason. Okay. Any doubt? Okay. I think so.
Dr. Chala sir to honor us with his delightful presentation. We came to know, and we and our students came to know a lot about vaccines, animal models, and their workings, their challenges. Thank you very much for this wonderful talk, sir. And I thank you to Mr. Sahu sir for your encouraging talk to our students for their future, for the roles of the pharmacist. Thank you very much. And most of all, I thank all the attendees here to to make this session successful. Thank you very much and have a wonderful day ahead. Thank you. We are closing this occasion formally. Thank you.